Hello, my name is Emmanuelle Vire and I work at the Institute of Prion Diseases in London in the UK, and I'm going to tell you about the projects that we are running in the lab and how we use epigenetic to study CJD. So in particular today what I'm going to talk about very briefly is the role of DNA methylation in diseases. I'm going to tell you what DNA methylation is, what it does, why we think it's important to better understand CJD, and how we investigate the role of DNA methylation in CJD. DNA methylation. It's easier to understand it if I take this model of a DNA molecule. As you are fully aware, DNA is made of 4 bases, which are here in 4 different colors. Only one of them, the green one, or the cytosine, can be modified chemically. This modification is what we call methylation. It is the adjunction of a methyl group to the cytosine. It comes in, but it can also be removed, so it's quite dynamic as a process. It does not involve any cut in the DNA molecule, so no truncation, no mutation. It is what we call a mechanism that comes on top of the genetic, and therefore in Greek on top of means epi, well we said epi, and so here we're talking about epigenetic, above genetic. What does this addition do? Well, we know that it strongly regulates gene expression. In other words, if a methyl group is present on the cytosine, this stops this region of DNA from being read or expressed. So it's a strong stop signal. Is this important? Yes, it is. It is what makes your cells being very differentiated. So your genetic code will be the same in all the cells of your body, but your cells perform different functions depending on where in your body they will be. For example, the cells at the top of your head are producing hair, but they're not producing muscles. So the chapter of the book, genetic book, that encodes the muscle gene can be silenced. And the way to achieve this is actually to deposit these methyl marks on the DNA that is responsible for the muscles gene on top of your head, and vice versa in other parts of your body. So DNA methylation is essential in maintaining cell differentiation, and that is when everything is normal. We also know that DNA methylation is very important in the way you respond to the environment. So should you go into the sun and your skin starts to get darker, at least for some of us, not me, but this response is actually these changes in epigenetic modification that very quickly respond to the environment. There are many more examples about the role of epigenetic and modification of the DNA in response to the environment, but this is key in this normal process of gene regulation. We know in disease that these profiles of DNA methylation are completely perturbed. And it is very important in cancer biology, I'm a cancer biologist by training, to look at where these marks are, because based on the profile of a genomic DNA methylation signal, we can first identify what type of tumor we are facing. Second, predict how well the tumor will respond to treatment. And third, and mainly, design new drugs to better target these tumors and these cancers. And this has been exemplified over the last 20 years because the FDA has actually approved 7 molecules, 7 drugs that can be used in patients and that are targeting these epigenetic modifications. So having said that, you now understand why it is so important to us to look at DNA methylation in CJD and better understand the role of DNA methylation in the pathophysiology of CJD. And so this is what we've been doing over the last 5 years. We've taken bloods from patients with sporadic CJD and compare their DNA methylation profiles or signatures, so where these DNA methylation marks are on their DNA. So we've compared that to healthy individuals. And we identified a very novel signature or fingerprint of DNA methylation that is entirely specific to sporadic CJD. We want to take this now further, so one step further. The first question is, how does this fingerprint compare in other type of CJD, in particular here in inherited prion diseases? And we have in London collated a lot of samples from what we call converters. So these are patients with inherited prion disease, and we followed them before and after they converted. So using this fantastic sample resource, we can now look in their samples, in their blood, where these DNA methylation marks are, and see how these DNA methylation marks evolve through time, but also before and after conversion. So basically, we've got three questions here. The first one is, can we find a signature that is different between before and after conversion in terms of DNA methylation? The second question is this signature, this fingerprint, different from other diseases? Can we yet again identify a fingerprint profile for DNA methylation that significantly is different from sporadic CJD and other neurodegenerative disorders? And then finally, and probably most importantly, can we identify factors from the environment, for example, that make conversion happen? You've seen that epigenetic response to the environment. Is epigenetic related to conversion, and if yes, how? That is probably the biggest question. So these are very much the three expected outcomes of this project, and I know we'll leave you with the most important slide of this talk, the one where I have to acknowledge everyone who's been supporting this project. First of all, the people here in London working with me, and more specifically, the huge support I've always received from both Professor Simon Mead and Professor John College, and then mainly from the CJD Foundation and all of you in the audience, the Robert Dodd Memorial Grant, the John Gill Memorial Grant, the Cheryl Molloy Memorial Grant, and the Marsha Snively Memorial Grant. Thank you very much for your attention. Please do get in touch if you have any question about the project, or if you want to know more. I love what I'm doing, and I always like to talk about it. Thank you very much.